Today we're going to be talking about incomplete dominance. So we've talked about regular dominant and recessive problems, and now we're going to introduce a new type of problem. If you have your note packet or notes, there's technically not anything you need to write down just yet. I will let you know if there's anything you need to write down, but for now, just focus on what I'm saying. So in regular dominant and recessive, we have capital R is red. If they have a capital and lowercase, it's also red. Two lowercase makes white. In incomplete dominance, what we have is we have two different letters, and that's because we've got two different genes. Neither of those genes is really dominant over the other, but what happens is we have a dominant and a dominant, or a dominant and a different dominant, or two different dominants. So in this case, they blend to form an intermediate trait. Some examples of dominant, incomplete dominant traits. Here, waviness. So we've got curly hair, we've got wavy hair, and straight hair. So somebody with straight hair gene marries somebody with a curly hair gene, and they get somebody with wavy hair kind of in the middle. Some examples of coloring, like dark purple versus white. They blend to make light purple. And horses. White with red oftentimes leads to a goldish color horse. So the question is, how do we do these types of problems? So when we do these types of problems, we are going to start with the first step. As always, determine what trait's dominant and what is recessive. This flower color is incompletely dominant. When a red and white flower cross, a pink one results. So in this case, we technically neither trait is dominant over the other, so we're just going to say that they're both dominant. So we've got red and we've got white for dominant. Even though we said, hey, technically neither one's dominant over each other, we're just going to say they're both dominant, and they blend to make pink. Step 2 is assign the letters, right? So for red, we are going to assign an R. And remember, each parent has to have two letters, so it'll be big R, big R. White will be WW. The blended one is always going to get a mix of the other two letters. So we're only using two letters. Don't do a P for pink. We're only using an R and a W in this case. Then we got to figure out the parent's genes. So it says the one flower is pink and the other one's white. Pink is going to be RW and white is going to be WW. The rest of the Punnett square is the same, okay? So we have RW, WW, one letter in each sperm, one letter in each egg. We're going to put them around the Punnett square, fill it in, and then we determine our percentages, right? So here we have WW, that's 2 out of 4 are going to be white, and RW, 2 out of 4 are going to be pink. So now that we know what incomplete dominance is, we're going to go ahead and fill in our chart in your notebook or in your note packet. So we know that in incomplete dominant, two genes blend. And actually, I'm going to add in here, neither gene is dominant and the two genes blend. I have an example of the genotypes and phenotypes here, right? Big R is red, big R and big W is pink, and WW is white. And then some of the examples we went over are coloring and then hair curliness. In terms of diseases, there are only a handful of diseases that kind of show this pattern. You'll remember with cystic fibrosis, somebody who was big A, little a, had enough normal receptors, so they were normal, right? They were still the dominant normal gene. With incomplete dominance, somebody who's in the middle would show some of the disease, but not all of it. And so one of the examples we have here is familial hypercholesterolemia, which just means genetic high cholesterol, okay? And you'll see somebody who's got two big A's, they have normal cholesterol levels because they have this receptor that can bring the cholesterol into the cell where then the cell can get rid of it. Somebody with two little a's has lots, really high cholesterol levels because the cholesterol can't get into the cell where it can then be turned into other stuff. Somebody in the middle has somewhat elevated. Notice they don't have quite as many receptors, and as a result, they do show symptoms of somewhat elevated cholesterol. So it was where in regular dominant and recessive, they might have enough channels and they don't show symptoms. In this case, they don't have enough of those receptors and they do show some symptoms, but not as many as somebody who is homozygous.